Hi, I am Dr. Ravikiran Yedidi. I'm the founder of the TCABS-E. So we started TCABS-E in Rajamahendravaram as a headquarters, and recently we actually started a branch in Visakhapatnam, and I'm actually quite happy about this because Visakhapatnam has a huge student base. Uh, the goal of TCABS-E is to bring in the international standards in training, especially the biomedical research arena. As most of you already know, the IT versus BT, the information technology versus biotechnology, India had made a tremendous improvement and progress in the IT, the information technology. I'm sure there are plenty of software engineers and a lot of people who are not engineers are also getting into the software because you could quickly learn a package depending on the market demands and you just get a job right away. However, that's not how it works in BT, unfortunately. Biotechnology is all about, identifying a problem. You have to come up with a hypothesis and design the experiments. And, you do the experiments and hope to get the results, the anticipated results. So these are all time-taking processes. I've nothing against the IT, but biotechnology is actually doing its best, especially with the Indian Institute of Technologies and Indian Institute of Science spreading all over India with their branches. I'm quite happy to see there is a pretty good improvement in the biotech arena in India compared to 20 years ago when I got my master's in biotechnology from India. In addition to this, we want to take this to the next level by bringing in the cutting edge technology. So a lot of students, they are, very proactive, and I have a PhD in biochemistry and molecular biology. Before that, I got a master's degree from Andhra University. So I actually got the master's from Yedidi College, which was back then affiliated to Andhra University. But when I got my master's in biotechnology, I didn't really think that I had all these skills, the practical skills to actually take up the biomedical research, which I was always passionate about. But this is something that actually bothered me for quite a while, but fortunately, I'm actually happy that I got a chance to go abroad, and I got a master's degree again in the United States from Wayne State University, Detroit, Michigan. So my master's in Detroit, it's molecular biotechnology, and this was my first practical encounter with genetic engineering, and obviously I was really excited about it. I learned anywhere basically from plasmid extraction from bacteria to PCR, site-directed mutagenesis, DNA sequencing, and basically the genetic engineering related stuff. I did a summer project related to mosquitoes, where one of the professors back at Wayne State University was actually working on West Nile virus. So I got an opportunity to work on this project where I would be screening the genomic DNA from mosquitoes to actually identify the odor-binding protein coding gene, which was my first kind of the genetic engineering project. As I mentioned earlier, I didn't have enough practical skills, but I was lucky enough that I actually got to work in my advisor's lab before I got into this genetic engineering project. So I actually built up a few skills like PCR, gel electrophoresis, DNA sequencing, and so forth. But that was like me taking a stab right up front. But it was okay, I survived. I finished this project and I finished writing the report and I actually got a good grade. And when I actually think about it, so this was back in 2001, so now 2020 is almost 20 years ago. When I think about this, I don't want the future generation kids to go through the same experience because they should know the practical, they should have the practical exposure. They should have the exposure to the practical skills, and also they should build these practical skills so that they don't have to step back and think twice before taking on any of these biomedical research. Could be a simple mosquito project talking about West Nile virus, or it could be a complicated HIV drug discovery project, which was something that I worked on in my PhD. But this is where TCABS-E comes into play, because I wanted to bring that skills back to India. As we all know, the Indian brains are smart. All we need is the proper guidance and directions. So I believe that if we can bring that technology back to India and guide the students, maybe from the undergrad level, including the postgraduates, that actually helps a lot, so that we can actually have these undergraduate students and postgraduate students focus on the biomedical research, different kinds of projects, and so without any hesitation, because this is what we've been going through. And if we can actually bring those skills to India, and if the undergraduates are able to perform lab practical skills in par with the undergrads in the United States, then I feel good about it, because that's what I want to see here. Because I worked with several undergraduates when I was in the United States and Canada, and they have worked on different things, anywhere from simple basic cloning, recombinant DNA technology, protein expression purification, cell culture experiments. And if they can do it, we can do it here. So I'm bringing that logic back to India to see, why can't we empower the undergraduate students and postgraduate students with these practical skills? And this is what is needed. I mean, that's what I believe anyways. If we want to bring the biotechnology or the biomedical research in par with the information technology progress that India had made. And I mean, India is also pretty big in pharma in comparison to the world pharma. As you all know, that India is actually one of the leading pharma companies that is that had supplied a lot of drugs in the past, and including the latest hydroxychloroquine during the COVID-19 pandemic. Typically, students have been thinking of jobs in pharma, but oftentimes what they don't understand is there is the pharma and there is the biotech, so there are two different things. For pharma, it's more of production, where you do a simple chemical reaction or multi-step synthesis of a drug, and bulk production of these drugs. Versus biotech is where you actually need the skills. And I'm actually glad that there are several biotech industries, companies in India compared to 20 years ago. And these biotech companies, they need special skills, like anywhere from basic cloning, DNA agrocellular electrophoresis, STS page, maybe HPLC for protein purification. For example, if a company is looking for people to help them in vaccine production, obviously you should be able to perform the protein production technology by yourself, and that's when they will actually take you into those kinds of jobs. So this is something that I always try to clarify to the students. It's like pharma and biotech, they are industry, but they have their own different signatures when it comes to the skill sets that are required to get a job into these industries. With that being said, I completed my PhD in Wayne State University, and I moved on to a startup company in Ann Arbor, Michigan. So that startup company was actually looking at some molecular diagnostics for cancer biology. And that's when I actually realized, so I was placed as a technician in that startup company. But when I realized that, as a technician, I was just doing PCRs on a regular basis to genotype mice that are, from the in-house animal facility. That's when I realized, I don't think I wanted to spend the rest of my life doing PCRs, like 5, 10 PCRs or 20, 100 PCRs a day. That's when I actually realized that I should be going back to the grad school and work on my PhD, where I can actually apply my thoughts and ideas, and where I could go creative, and that would actually give me endless opportunities to come up with new experimental procedures, or come up with new products, or even new products, patents, etc. So that actually prompted me to go back to grad school. So I went back to my PhD, and so I got my PhD in biochemistry and molecular biology. So during my PhD training, I was extremely lucky because my PhD advisor, Dr. Ladislav Kovari, gave me enough independence that I could actually design and work on an interdisciplinary project. So my project started with a basic biochemistry experiment where I was just doing protein estimations, standard BSA curves, etc. But the protein that I was working on was HIV-1 protease, and the specialty of that HIV-1 protease was actually it was an isolate from a patient who actually failed all the drugs. So this patient from which we got the sample didn't have any choice. So all the FDA-approved drugs that are available in the clinic are useless, because this patient had developed an HIV strain that is highly mutated, and it's resistant to literally every drug that is out there in the market. So this was back in 2004. That's when I actually got a chance to work on this clinical sample, clinical isolate. I was lucky enough to actually learn how to purify the protein. Although I started with the knockout version of this protein, because protease cleaves itself, so we have to be smart to actually protect your protein from your own protein. So eventually I actually mastered the technique of protein expression purification. So starting off, I was actually lucky enough to solve 4 crystal structures of these isolates in the first semester of my PhD, and which actually resulted in a publication. And that's when I realized it's like PhD is the place where you can actually apply your creativity, your knowledge, your skills, and come up with innovative things. And then when somebody solves 4 crystal structures and analyzes them, and that's pretty much half the PhD project. But I didn't feel like stopping it there, so I asked my PhD advisor if I could go further and do some drug discovery. And my advisor, Dr. Kovari, was actually nice enough to give me the freedom to go and collaborate with Pfizer. And so I came in contact with Dr. Ed Pagani from Pfizer Ann Arbor, which no longer exists. But back then Pfizer Ann Arbor was pretty functional. So when I spoke to the scientists at Pfizer, they said, drug discovery is definitely the way to go. And so I've actually redesigned my entire PhD project in such a way that, it reflects the full drug discovery platform, all the way starting from choosing your drug target to the final product that gets tested in the animals in clinical trials. So during the process of the 4 years from 2004 to 2008, I was able to actually do crystallography, NMR, computational biology, of course molecular biology, biochemistry. So it's a combination of all these things. So that's how my PhD became an interdisciplinary project. So at some point when I finished analyzing the crystal structures of this patient isolates, I asked my advisor if I could go ahead and actually design new drugs that would actually inhibit this patient sample, which is resistant to all the drugs. And I was lucky enough that I got the permission. So I learned the computational biology, and I've actually designed peptidomimetics based on the structure of the substrate-bound HIV-1 protease, and which eventually ended up being my PhD thesis. So it's basically structure-based drug design. And the peptidomimetics, we came up with a library of 6 peptidomimetics, and then my advisor told me that's pretty good for a PhD, but then I said, can I go ahead and synthesize them and evaluate them? Which he wasn't sure about, because he wasn't sure about my chemistry skills, my organic chemistry skills. But then we collaborated with someone in the Wayne State Pharmacy School, and luckily I got a chance to actually go and synthesize these peptidomimetics. And some of them are 16-step synthesis, so that was hardcore synthetic medicinal chemistry. After 1 year of hardcore synthesis, we finally got about 5 peptidomimetics synthesized, and I've actually co-crystallized them with the isolate, and then we've actually solved the crystal structures, and we also evaluated them biochemically. And so we've got a lead peptide, or peptidomimetic, was about sub-nanomolar range, and so the initial hit was about 4 nanomolar, so which was pretty good based on structure-based design. And so that actually gave me a full confidence that I can actually do drug discovery, and it is it is a very complicated technique, but I could do it. If I could do it, anybody can do it. That's when I thought, maybe I should just train people in drug discovery, and that's how we actually came up with the drug discovery program in TCABS-E now in India. So if anyone wants to learn how to design drugs, all the way starting from a drug target, a protein structure, analyze the binding pockets, and design a compound, and synthesize, evaluate biochemically, and also in vivo. And I was actually lucky to do some in vivo studies also, because I was a part of the systems biology workshop that was conducted by the Michigan State University in Lansing, Michigan. And during that workshop, we've actually learned how to do dose response curves using rats and mice, and also we've done quite a few surgeries actually, and I'm actually happy that the animals survived my surgery, which is often a big deal. And so that pretty much completed the preclinical drug discovery. So my PhD, the entire PhD training can be considered as a preclinical drug discovery that goes on in industry, whether it could be biotech or pharma. So this is something that I was always interested ever since I was a kid. I wanted to know how the drug discovery happens. And when I got that skills, I thought, I should probably train other people in this, because this is really cool and interesting. So now we actually have a 2-year certificate program in TCABS-E that focuses entirely on drug discovery. So if anyone is interested and curious to know how the drug discovery works, you're more than welcome to come over and we can discuss the possibilities. And one thing that I've noticed is people, often students, think that they may not qualify. My suggestion to you is, don't pull yourself back. If you think you're not qualified to do drug discovery, just come and talk to me and see where the discussion goes. Let me ask you a few questions and see what you're passionate about, and let's see what skills you got. And if still you think that it is not your cup of tea, of course, you can always walk away. You have nothing to lose, except for 5 minutes conversation with me. So this is very important, because I'm not here to push the practical training onto the students. The student has to come with enthusiasm, and the students have to have that passion to actually learn. So that's why we're not really big on advertisement. I'm sure you've probably never seen me or my TCABS-E in the TVs or in the movie theaters, because we're only interested in students who are passionate, who are interested in learning science and doing science and making a difference in the society and in the the life of humans. So if you think you have those qualities, definitely I'm open to talk to you, and please come over, and I'll be glad spending time with you.